What they did was they took the frog embryo, they slice, and put all the little slices onto your slide. So if you're looking at one and it doesn't look quite right, you might slide on over to the next one, and you'll just have a slightly different view of it. Now, this is the only one I have, and I don't really like it. This isn't quite test quality. But one thing that you can see on this is that so here was the original egg, and you can see that there are smaller blastomeres here and large blastomeres here. And what that's implying is that here there was a plane of cytokinesis, let's say, went this way, and a plane of cytokinesis that went this way. And then the third plane of cytokinesis was perpendicular to the first two. It's cutting across differently. But notice how it's been displaced way to the top. So you get this disproportionately large cells down here and smaller cells up on top.
That's kind of like one giant piece with maybe like a cut, two cuts through it. But
do you have another section on that slide?
Yeah. Yeah, they're all really solid pieces.
Okay. Well, the reason that you get smaller cells on one side is because there's less yolk. When this egg is sort of hanging in the water here, all the yolk settles down to the bottom. The side that hangs low with yolk is called the vegetal pole, and the opposite side is called the animal pole. Because the yolk has settled down to the bottom and cleavage was hard through the yolk, then the third cleavage line was displaced upward, where it's easier to cleave. You'll also notice, does that make sense to everybody? So at this stage, I could already orient an animal pole from a vegetal pole. And I can kind of see like, oh, these are going to be smaller blastomeres. These are going to be larger blastomeres full of yolk.
Animal pole?
Animal pole versus vegetal pole. You're just taking a globe and you're trying to give it coordinates. You'll also notice maybe, and mine doesn't really have it, across the animal pole will be a dark pigmented line. See if you can see pigment.
Yeah.
It'll be across the animal pole.
It's a little thicker.
It's the line?
Yeah.
It's dark. I mean, it should be black on your slide.
Yeah.
And I'm seeing a bunch of heads nodding, so
Yeah,
presumably you guys are getting it. Now, the pigment will be across the animal pole. It'll be draped over the animal pole. Why? We're not quite there yet. But why, even before sperm, there will be a pigment layer over the top.
Oh.
Shade. It might be protecting the nucleus of the egg from UV light. What else? Counter shading. What's counter shading again? If you're a fish swimming over the egg and you look down, you see black against a black pond bottom. If you're swimming under the egg and you look up, you see white against a white sky. This is counter shading, and you'll see this over and over in the animal world. I mean, like all the way from my tabby cat, who has much darker back fur than belly fur. You'll look at a shark, much darker on the dorsal than on the ventral side. Counter shading is all the time. You'll see this in the animal world all the time.
Does it also help with keeping the egg oriented up?
The pigment won't help with keeping the egg oriented the way it is, but the yolk settling down to the bottom creates an orientation. Now, what's another thing about putting pigment on the top of the egg? Black pigment. That was a bit big. This is a solar collector, and you are warming the egg this way, increasing the possibility for successful development. All right, so there's a few little things. Now, it is true that from the point of sperm entry, the pigment rotates toward the sperm entry point. But I'm not making a big deal out of that. I really want you to get the idea of counter shading, warming, UV protection, animal pole, vegetal pole. Lots of yolk down here, smaller blastomeres up here. Cool. Next slide. Here is small blastomeres along the animal pole. I am still seeing a little bit of that pigment up along the animal pole. Big cells with lots of yolk down here. The blastocele right here. There's not a whole lot, and that's why I say 15 seconds. Any takers? We're done. Go to the gastrula. You're seeing a layer of cells out here with very small nuclei. Small cells out here. That means that they came from the animal pole. As the cells of the animal pole continue to divide and push against each other, they're sort of pushing cells down around the vegetal pole. The animal cell poles are dividing faster than the vegetal cell poles. And they start to migrate downward around this. There's sort of a movement down around this ball of cells. And you can see that right here. These small cells moving down here toward the yolk plug right here. And then they kind of, in a sense, they sort of like push against each other. There's nowhere more for them to go, so they fold back on themselves and start coming up on the inside. Look at your slide and see if you can visualize what I just said. Cells migrating from the animal pole toward the vegetal pole, hitting the yolk plug at the bottom, and then curling inward and starting to migrate upward on the inside. Once you get that, that's your folding. That's what creates a gastrula. And so at this point, I could identify an ectoderm on the outside, a sort of an endoderm coming up here, and in between the two, a sort of a mesoderm. See how here's the endoderm, and here's a mesoderm. Ecto, meso, endo. You created the 3 germ layers when this thing folded in on itself. What's this down here? What's the opening? Blastopore becomes the plug. Becomes what? Anus. Becomes the anus. Okay? And then somewhere, here's kind of an archenteron of sorts, endoderm being made, and somewhere there will be a mouth that pokes out somewhere else, deuterostome, somewhere other mouth. Okay? Now soon the archenteron, and in fact I think I'm kind of getting faked out. I think most of what I'm seeing in here is archenteron and the blastocele is being pushed out. Some of you guys will see like a nice clean blastocele on your gastrula. Some of you will maybe be seeing some of the archenteron already. Now what you're seeing here again is ectoderm. Let's come around. This isn't showing the yolk plug or the blastopore, but that endoderm has come up on the inside, and the mesoderm is a whole strip like this. Notice this little circle, perfect circle up there. Find a perfect circle if you can. Wave your hand. The mesoderm has migrated up inward here, and a little bit of mesoderm broke off from this side, and a little mesoderm has broken off from this side and made this. Now remember, what you're looking at is a whole ball of cells. this is what's hardest about this exercise, is you've got to do this mental calculus and see this in 3 dimensions. So you're looking through a slice here. Remember, this round thing right here goes all the way down the back of this organism. This is the dorsal side of this organism. This then is the belly side of this organism. And the frog embryo, the little tadpole, is facing toward you, and I've chopped this guy. And so here is his notochord, or what will be sort of the vertebral column of this guy. Remember, bone, vertebral column, is made of mesoderm, and this mesoderm here, making the notochord, is given a special name called chorda mesoderm. I'm not asking you to write that down. I'm asking you to look at this, and at this point, technically, it's given a different name, but in the near future, we would call this the notochord. So you just learned it as notochord. So here is mesoderm, kind of coming up, mesoderm kind of coming up. There's endoderm and endoderm here. Mesoderm, and then ectoderm. Clearly I have my 3 germ layers. Endo, meso, ecto. I have the beginning of a notochord. And I suppose at this point I should mention what a chordate is. The phylum Chordata has 5 major hallmarks. And the first one is the presence of a notochord at some point in its development. And I'm spelling this with an H, and I'm not sure if notochord is C-H-O-R-D or C-O-R-D. But a C-H, yeah. Oh, good, I got it right. A notochord is connective tissue. It's like a thick, stiff connective tissue rod that at some point in your development you had one. And some organisms, that's all they ever get, and they keep them their entire lives. Some of them have a notochord when they're a larva, but then lose them as adults. Those are very, what we would call primitive chordates. There are other hallmarks, like pharyngeal gill slits. Pharyngeal, pharynx means around my throat. Gill slits means little openings. And, believe it or not, maybe you guys have all seen the photos, at one point you had gill slits. They have grown over, and those have become other structures that are like your inner ear bone or middle ear bones and things like that. Those gill bars and gill slits became other structures, but you had little gill slits at one point. We also all have what's called a post-anal tail. And if you look at the skeleton of a human, you see a little post-anal tail here. There's 3 or 4 coccyx bones. Did I tell you about one of my old girlfriends who was born with a little tail?
Mm-hmm. Shallow Hal.
What's that?
Like Shallow Hal?
Shallow Hal had a tail?
Yeah.
I don't know the movie reference, I'm sorry. But they, you know, and this is not unusual to see people born with extra coxal vertebra. And what they did was they just lopped them off right at birth. They just chopped them off. And, you know, it's like she never knew it, but there was a little scar there. I just thought it would have been kind of cool, you know, like have a tail, you know. But they thought it would be hard for her to buy Levi's and stuff like that. But you could always tell if she'd been bad, her tail would be down, or if she was happy, her tail would be down. Let's see.
Oh, right.
What am I forgetting here on these guys? We all have, a dorsal, and this is where I'm going here. A dorsal hollow nerve cord. Now remember that protostomes, all the invertebrate guys that we looked at, have a solid nerve cord that runs down their belly. We, on the other hand, have a nerve cord running down our backside, dorsal, and it's hollow. And there's a reason that it's hollow, and we're about to figure that out. There's one other feature of chordata that kind of has only been tacked on in the last, I don't know, couple of editions of Hickman, and that is something called an endostyle, which some organisms, and you'll look at these organisms that have it, have this ridge that runs down inside of their throat that secretes mucus. And that mucus traps food. Okay, now what we have done with that endostyle, okay, you've got a ridge of tissue down your throat here that secretes stuff. We have an organ right here on our larynx that secretes stuff. What's that organ called? The thyroid gland. And so some chordates have changed their endostyle into a thyroid gland. But these are, if you have these at any point in your development, you're a chordate. You're in the phylum Chordata. These are what we call the hallmarks of the phylum. They are the key features. Now, we can see the beginnings of the notochord right there. Now, we can also see the beginnings of the dorsal hollow nerve cord right there. So let's start to follow that. In fact, I'm going to do that on another board.
Did you say you needed all of those?
Like this. And good enough for me. I said that this was the notochord. And what you start to see here is that the notochord starts to secrete growth hormones upward. And you're seeing this rapid division just above the notochord. And that's why this is starting to pucker upward like that. And that's the beginning of the nervous system. This is called the neural groove here. And what you'll see is, I'm just going to draw the top part of this. Here's my notochord, and it's still secreting, and it's secreting and causing rapid division, and these guys eventually seal, and this whole thing becomes my dorsal hollow nerve cord. Well, and you will see on the next slide how these two little humps keep growing and growing and growing up and touch each other and fuse at the top. But they fuse around a hollow cavity. We'll see that. There's one other thing on this slide, though, that I want to point out. Do you basically see the direction that you're going from this slide to a dorsal hollow nerve cord, first of all? And we'll look at the next slide, and that'll help confirm that. The next thing that you're seeing here is flanking the notochord are these two sort of long triangular strips of tissue here and here. See if you can find those on yours. There should be two sort of skinny triangular strips of tissue flanking the notochord. Yeah, I have a really crummy neural tube slide. I've got it on the best section I have, but it's not as good as yours probably. This is showing that the neural groove has come up, fused to itself. There's the little hollow that runs completely down through my spinal cord. Through your spinal cord, is a hollow channel full of cerebrospinal fluid. Now, I never mentioned what somites are about. I just wanted you to first identify them. And on this, I can see here's a triangular strip, here's a triangular strip. Somites are mesoderm. What does mesoderm create? Lungs? Organs? Yeah, maybe some of the big squishy organs.
Reproductive.
Muscle and bone. The somites create, along these strips, create the muscles and bones of those strips. Somites create the muscle and bone along that particular region. Now remember that by this time, Hox genes have already started to identify segments. And when you're looking down a vertebral column here, you can see, oh, look, right here, I need to create this rib. I need to create part of this scapula. I need to create, all the way around, I need to start creating the muscle and bone of this strip here, of this strip here. One of the problems with humans is we got these really long legs and arms, and so when you're seeing me, you're seeing really all legs and arms. Here is my body. This is my body right here. It's standing way up on top of these long legs. So this is the part that I'm talking about. Legs get created by what toolkit protein? Say it again? Distalis. Okay, so that created this long, kind of weird thing that projected out, which we call legs. But here's my body, and here's where I'm segmented. This is the segmented part of my body. And so I have these segments that are sort of created by somites. Okay? Now we make, so what you've gone through up till this point is from a fertilized egg, basically, up through the creation of a nervous system. The spinal cord is the basis of your nervous system. You're also seeing the basis of all your bones and muscles per strip. Now we go and we make this huge jump, and I've got other slides, but I just don't want to beat you up with slides. I probably already have. So what you want to do now is go to the 10 millimeter frog sagittal section. SAG, sagittal section. And what that means is you're going to be looking at a little froggy, and what they've tried to do is they've taken one tadpole and they've gone like this. Chop, 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 chop. And then they've laid all those little sections down on there. Now in some sections you're going to see an eye, and in some sections you're not going to see an eye. So the presence or absence of an eye, and right here is a good eye, is going to tell you kind of where you are on the tadpole. Am I out toward an edge, or am I right down the middle? And what I wanted to show you with these slides is kind of maybe some of the rest of the hallmarks, as well as the development of certain organs. So here on this, I can see an eye. So I know I'm way out on the edge. Am I going to see a whole lot of nervous system way out here on an edge? If I cut right through the middle, I should see not only the middle of the brain, but also the middle of the spinal cord. But way out here on an edge, I'm totally missing the spinal cord, and I've got a little pocket of brain out here. But what do I have right here? The mouth is actually going to be right about here. But remember, I'm way out on the edge. What am I seeing out here? Ooh, those are the pharyngeal gill slits. You see how there's like bars right there with little water channels to come through? Here's my pharyngeal gill slits. That was one of the hallmarks of courting. Now in the end, a frog doesn't have gills. After metamorphosis, they don't have anything to do with gills. But early on in their tadpole existence, they do have gills. And early on in your tadpole existence, you have gills. We can't rewrite our genetic program from scratch. We don't need gills. We never did need gills. But we can't just say, you know what, we're done with that. We're amniotes. We're done with all that gill stuff. You can't just fundamentally rewrite everything. And so, to some extent, the development that you see sort of tells a little bit about your evolutionary history. I don't want to get too dogmatic about this, but you do see a sort of an evolutionary history written in our development. The fact that we once had, in your development, you once had gills, means that we as a phylum once had gills. And some animals still possess them, like fish. Now going back on this little organism, I'm going to see if I can jack this thing around a little bit. Well, there's something that I can talk about, but I'm going to go to a different slice first. Now I've moved a little more closer to the middle, cutting right through the middle of this tadpole. And up here, now you can see his mouth really clearly, and through the pharynx. And then I kind of lose track of the pharynx. Maybe that's it right there. And maybe I'm coming back through the intestine as I go back. Here's this dorsal hollow nerve cord coming up into his brain up here. And I can see brain tissue. Really what I'm seeing here is the hollow parts of the brain. Do you remember what the hollow parts of the brain are called?
Ventricles.
The ventricles. And I'm seeing the hollow part of the spinal cord and the hollow part of the brain up here. I'm seeing the mouth. And now I'm coming back. I'm coming down through the mouth, and I'm coming through the heart. I want you guys to find the heart sort of in the middle of your tadpoles right now.
Can you see the three chambers?
You can even see the three chambers. 2 atria, 1 ventricle. You can see little red blood cells in there.
You see? You see? I'm going to take the camera and
I'm pointing inside.
And those two big ones.
Numbers on this? Absolutely not. But if you get a really good slice, like Tyler's had, well, maybe you can see it. I don't know. Way up here is the head and heart, and I'm going down, 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 down. I'm looking through. Here's this woven up intestine. I'm going down, 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 and here I'm seeing the intestine coming out, and there's the anus right there. And then, what's behind the anus? The post-anal tail. You have all this as the tail behind the anus, post-anal tail. So there's one more hallmark of the chordata. I'm also seeing, and see if you can find, here's this strip that goes all the way from end to end, and it looks really almost like clear, with little cross, see there's just a little bit of cross members between there. See if you can find a real clear strip or line through your critter. You have to be on a pretty central cut. And I'm watching this going all the way up through. It's a really clear, compared to everything else, it looks very clear. That's the notochord, and that's a great landmark for you. Well, it's a connective tissue. It's cellular, but connective tissues secrete extracellular matrix so that there's some collagen and a lot of elastic fibers at this stage, more collagen later on in the organism's development. Muscles can attach to a notochord. The notochord eventually breaks up in us. We ossify some regions into vertebra, but the only thing that's left of your notochord is the intervertebral discs, or the little pads of connective tissue between vertebra and vertebra will be a little spongy disc. Your notochord, the remnants of your notochord that made up your intervertebral discs. That's all that's left.
Can you point to the notochord on your...
Sure, it's this clear zone that runs right down his back. Above the notochord, remember, must be the spinal cord. Below the notochord will be all the guts of the animal, and the notochord itself you could think of as a sort of a primitive vertebral column.
What's on the outside of I just forgot. Right on this. Yeah, what's that?
Well, okay, so if here's notochord, here's spinal cord, and then here would be what muscle or little fin-like things project off of a tadpole's back. There is a bit of a fin on the back of a tadpole, and you'll see that in cross-section. You don't see it quite so clearly here. Now, I do want to get out to an edge, and I know that this is probably driving you guys crazy by this time. This kind of slide work, I know, is hard. I want to go back to this one here. This is more out toward an edge. You know that because you're seeing an eye, you're seeing pharyngeal gill slits. You're seeing something else tucked up against the back. Remember, here's notochord, that's kind of that clear zone. Spinal cord. And notice if you can find anywhere where you see tube, a whole bunch of tubes. These tubes are the basis of the kidney. They're called pronephric tubules. Pro, first or before, nephron. The pronephric tubules will eventually grow into what this guy calls a kidney. Their kidneys aren't nearly as defined as ours, but then they live in the water where they can just drink and pee to their heart's content. Remember, these guys don't have a diaphragm. That's a mammalian invention. I mean, you might not have remembered that. That's one of those factoids that you will want to remember for the next week's lecture. I ain't saying that. I mean, it might not even be a question, but that's one of those things we talked about